And we're into part 2 now. Now we're going to look at the actual exchange in the capillary. Okay. So the capillary is where we exchange gases, where we're exchanging nutrients, where we're exchanging the byproducts of metabolism between the blood and the tissue. The exchange occurs by 3 different methods. All right. And you need to understand this. First of all, diffusion. This is the most important way we transfer stuff. The lipid-soluble substances will diffuse through the cell membrane. Water-soluble substances diffuse through channels and slits, but diffusion from an area of higher concentration to an area of lower concentration. Filtration is where we're going to use the pressure of water to force molecules through this membrane. All right, and the pressure is derived from the contraction in that heart. Okay, and then we have osmosis. Osmosis is diffusion of water. So you have a solute, such as the plasma proteins, that creates an osmotic pressure, and that draws water into the capillary, and this opposes filtration. So we'll talk about that in a second. But basically, filtration, we're forcing molecules through the membrane, and as we move things, then we get an opposing pressure. So in terms of the osmosis, at the arterial end of the capillary, the water and the other substances are going to leave the capillary by filtration, all right, because the pressure's higher on that side. You have to think of the hydrostatic pressure as the pressure of water. Colloid osmotic pressure is the pressure of the stuff dissolved in the water. So as we are at the arterial end, you've got, because of the push of the blood, the hydrostatic pressure is higher, and we start to move stuff over the membrane. Okay? And as we move things out of the blood vessel, out of that capillary, and into the tissue, that makes the colloid osmotic pressure rise. Okay? And that makes the hydrostatic pressure within the blood vessel drop. So at the venous end of the capillary, the water gets reabsorbed back into the capillary, all right, because the colloid pressure is higher than the hydrostatic pressure. So more fluid leaves at the capillary at the arterial end than returns at the venous end. And what will happen is you have lymphatic vessels. We haven't talked about the lymph system, but the lymphatic system will return excess fluid through the lymphatic vessels to the venous system. Okay? And then we have transcytosis, which is where we're moving large substances, lipoproteins, antibodies, that sort of thing. And that's done by a process called endocytosis. And the substances in this are moved into the cell, and they leave the other side of the cell by exocytosis. And we wrap it around with the membrane, and then we take it out. So this is the capillary exchange. Okay, we're coming in from the arterial. Because of the pressure, the blood pressure, this is higher than the pressure in the cell. So things will move into the cell. All right. As you continue to move stuff into the cell, the pressure changes and eventually becomes higher over here, and you start to move stuff, wastes, out of the cell and into the capillary, into the venous side of this capillary. And that's really all it is. Okay? The venules are the microscopic, tiny veins that transport the blood from the capillaries to the veins. They have thinner walls. They don't have as much smooth muscle. And then you move into the veins, and these have thinner walls than the arteries. Again, you don't have the blood pressure in the venous side than you have in the arterial side. They do have 3 layers, but you don't have the muscle in them, as much muscle in the veins as you do in the arteries. And because they're carrying this blood under low pressure, all right, the venous system is your blood reservoir. Most of the blood in your body is stored in the venous system. One thing that veins have are valves. Veins have valves, and that keeps the blood from backflowing back the vein. Okay, it's one thing if you're above your head. It's another thing when you're below, not above your head. Yeah, right. Above your heart. It's another thing when you're below the heart. When you're down in your big toe, it's hard to get the blood all the way back up to the heart. And so what happens is as you push the blood up into the vein, you have valves that prevent this blood from backflowing too far. All right. So if you look at this in this distribution, you can see there's an awful lot of blood that's in the veins. It is about the 2/3 of the blood is in the veins and the venules at any one time. It is a blood reservoir. Okay. All right, some application here, atherosclerosis. Again, this deposit of the plaque, cholesterol plaques in the inner lining of the walls and starts to narrow those vessels. You may have an aneurysm, that's a bulge in the wall of the artery that forms when the blood pressure dilates a weakened area of the vessel, and it can rupture. It can thin enough that it leaks and doesn't completely rupture, or it can rupture. Okay, it's very common to have abdominal aneurysms, aortic aneurysms, brain aneurysm. Phlebitis is where you get inflammation of the vein. This is very common. It swells, gets red. And then varicose veins, very common in women that have been pregnant and older folks. This is where you get the dilation in the superficial veins. And it's just more common in the legs itself. All right, so let's talk about blood pressure. This is the force of the blood that the blood exerts against the inner walls of the vessels. The blood pressure usually refers to the pressure in the systemic arteries, but you have pressure throughout the entire system. Blood moves from an area of higher pressure to an area of lower pressure. So we have these different blood pressures. We look at arterial blood pressure, and this rises obviously when the ventricles contract, and it falls when the ventricles relax. So we have systolic pressure and diastolic pressure. The systolic pressure is the maximum pressure during ventricular contraction. The diastolic pressure is the minimum pressure remaining before the next ventricular contraction, so when the ventricles are at rest. The difference between the two is the pulse pressure. All right. Blood pressure is stated as a fraction. We've all heard it. My blood pressure is 120/80 or whatever. So it's the systolic pressure over the diastolic pressure. And we measure this in millimeters of mercury. And we use a blood pressure cuff, which is, you ready for this? The medical name for a blood pressure cuff is a sphygmomanometer. I've had that one in some trivia contests. So remember that one, sphygmomanometer, blood pressure cuff. All right, so we can see this blood pressure decreases. It's the distance from the left ventricle increases. So the further you get away from the pump, the further down the blood pressure goes. Well, that's a kind of like duh moment, okay? There are several different places where you can measure arterial pressure. Most of the time we're going to use the radial pulse and pressure, the pulse. We usually use the upper arm or the lower arm for the pressure, but it's usually the radial, but we can do a femoral, we can do a popliteal. There are several different pedal pulses to measure this. Okay, Some of the things that influence arterial pressure, blood volume, the viscosity, how thick it is. Okay, are you dehydrated? Are you overhydrated? Heart rate makes a difference. Stroke volume, how much is being pushed out of the heart, and then the resistance. Okay, so these cardiac output, the volume of blood that's being discharged from a ventricle each minute is equal to the stroke volume, which is the volume of blood that enters the arteries each ventricular contraction. So when the heart beats, how much blood is going out with the contraction? 70 mLs. And that's a ventricle. So the cardiac output, the stroke volume, times the heart rate, about 5,000 mLs a minute. Basically, what's in our blood volume that we talked about. So any increase on either of the components of the equation is going to cause an increase in cardiac output, increase the blood pressure. All right, so think about epinephrine that we talked about increases the cardiac output, and it increases the cardiac output by increasing the heart rate and the stroke volume. That's why epinephrine works. Okay. All right, blood volume, we've already talked about. This is usually about 5 liters. Blood pressure is directly proportional to that volume. Any change in the volume changes blood pressure. So if you have a hemorrhage or something like that, blood pressure decreases. All right, peripheral resistance is the force of friction between the blood and the walls of that blood vessel. And pressure has to overcome this peripheral resistance in order for blood to flow. There are several factors that can change that, but the bottom line is the vasoconstriction of the arterioles increases peripheral resistance, which increases the blood pressure. So that when the blood is pumped out of the ventricles, the arteries swell, and you can feel it in your artery. You feel your pulse. What you're feeling is that the artery swelling and that elastic recoil, and that's what sends the blood through the arteries against the peripheral resistance that's there. An important piece is viscosity. Okay, how thick is this blood? The greater the viscosity, is it syrup or is it liquid? Okay, the greater the viscosity, the greater the resistance. So your blood cells and your plasma proteins can increase the viscosity. There are diseases that increase the viscosity, make the plasma proteins more viscous. The greater amount of blood cells can increase it. If you have an anemia, it lowers the concentration of blood cells, so it lowers the blood viscosity, so it lowers the blood pressure. Okay? So any factor that alters the concentration of the blood cells or the plasma proteins alters the viscosity. So when we're talking about blood pressure control, blood pressure is a product of the cardiac output and the peripheral resistance. All right, so what you have is we've already talked about cardiac output, we've already talked about stroke volume, but stroke volume now is going to be the difference between what we call the end-diastolic volume and the end-systolic volume, okay? Now, what's the volume at the end of diastole versus the volume at the end of systole? So the end diastolic volume, the volume of blood in each ventricle at the end of diastole. How much is in each ventricle? The end systolic volume is how much is remaining in each ventricle at the end of ventricular systole. So how much is there when it's at rest and how much is there after it's contracted? Okay. So one of the things we have to look at is preload. Preload can influence the stroke volume. It's called the Frank-Starling law. And it basically says the more things are stretched, the greater the force of the contraction. You can see this if you take a rubber band and you pull the rubber band, okay? The more you pull the rubber band, the harder it wants to snap back, all right? And this is what happens with the heart. So the more the heart is stretched, the more the stretch in the cells, the greater the force of contraction. So think about the rubber band. So with this insystolic volume, there are several things that affect it. One is the preload. How much is the heart stretched? The strength of contraction, and then what we call the afterload, the force that the ventricles must provide to open the semilunar valve. Because remember, the semilunar valve closes because of the blood backing up in the aorta and the pulmonary trunk, which opens the valve. So in order to push the blood out of the heart, you've got to push against those valves. So that's the force that has to be exerted against those valves to open them up. This is one of the problems with high blood pressure, is if you have a high pressure up against that valve, it's harder to open the valve. Okay? So understand that. So some of the things that can control that, epinephrine. We've already talked about this. This is from the adrenal medulla. We've already talked about what can happen to our fight or flight. Peripheral resistance. We've already talked about that. Vasoconstriction reduces the arterial diameter, increases the resistance, increases the pressure. Vasodilation, the exact opposite. You have a vasomotor center in the medulla that regulates the diameter of these arterioles and therefore the peripheral resistance to help us control blood pressure. So there's a whole line of things that regulate blood pressure, and just be aware, it depends on what is going on. So one of the things that a lot of people have is hypertension. This is a basically long-lasting elevated pressure. There are many different causes. You can see and look at them: psychological stress, obesity, kidney disease, increased sodium intake, and so on. Can be a silent killer. There are no symptoms of hypertension. It contributes to the formation of atherosclerosis. It can lead to a thrombus or an embolism. It can lead to strokes. It can lead to TIAs. So there can be a problem. And treatment is usually some kind of blood pressure pill, and there are different kinds. There are ACE inhibitors, there are diuretics, there are sympathetic inhibitors, excuse me, that will do that. All right, so let's look at venous flow. This is one of the things that, one of the problems is we've got the blood in a venous reservoir, and we have to get the blood back up to the heart. And blood pressure decreases as the blood moves through this arterial system and into the network. And we want this to be slow because we need this to be slow because we have to have exchange at the capillary level. If we just run this stuff across the capillary, nothing gets exchanged. We don't have enough time. So we need to slow the blood down so that exchange can occur. But that results in very little pressure at the end of the venous end of these capillaries. All right. Now, how do we get this back to the heart? Well, there are several different ways in getting this back. First, we can use skeletal muscle contraction. These vessels particularly in our legs, again, we don't worry about the veins draining above the heart. It's below the heart we're worrying about. And we have legs, and we have skeletal muscle contraction in the legs. And as these muscles contract as we're walking, they are going to squeeze these veins. And that's going to move this up. And then so you walk forward, it squeezes it up, you come off the muscle, and it backs down a little bit. But don't forget, we have valves. So it backs down to the valve, and it will sit there. And the next step, it goes up a little bit further, back, a little bit further, back, a little bit further, back, and it gradually makes its way back. The other thing we're going to use is our lungs. As we inspire, that thoracic cavity increases. The pressure goes down. We have formed a vacuum. This is going to suck that blood up towards that heart. Okay? Another thing that you don't think about, but as you contract the heart and then you relax the heart, the blood that's right there at the vena cavas is going to be sucked into the atrium. That helps. Then we also can use vasoconstriction of the veins. That certain reflexes can cause contraction of the smooth muscle in the walls of the veins. So, again, the veins can act as a blood reservoir, and they can alter its capacity in response to what's going on. And you can actually see how this is done with this picture. So you're, again, shooting it up there, and the valve is going to make sure that you don't backtrack it too far. All veins, except those returning to the heart from the lungs, drain into the right atrium. The pressure in the right atrium is called the central venous pressure. And there are factors that influence it and alter the flow of blood into that right atrium. A weakly beating heart increases the central venous pressure. Any increase in the central venous pressure causes blood to back up in the peripheral veins, which can lead to edema. All right? Just understand that. So, exercise is good for the heart. It has to be a regular part of life. The cardiovascular system will make adaptations to aerobic exercise. Again, you can see the things that will happen. The stroke volume increases, the heart may increase, you get hemoglobin concentration increasing, blood pressure decreases, heart rate decreases. For exercise to benefit the cardiovascular system, the heart rate needs to be 70 to 85% of maximum, or 220 minus your age. So yes, the older you are, the less you have to do, but that's okay. It has to last for 30 to 60 minutes, and it needs to be 3 to 4 times a week. Okay, let's look at passive circulation. Pulmonary circuit, systemic circuit. Pulmonary circuit, again, to the lungs and back. Pulmonary circuit, right ventricle, pulmonary trunk, pulmonary arteries, and then we basically go through the lungs and then back into the pulmonary arterioles, the alveolar capillaries, the pulmonary venules, the veins, and then back to the left atrium. The whole idea is that the blood in the pulmonary arteries and our arterioles are low in oxygen and high in CO2. So gas exchange occurs in the alveolar capillaries. All right, the blood in the pulmonary venules are then rich in O2 and low in CO2. And then that goes to the systemic circuit that sends it out from there. And we go from the left ventricle to the aorta to all the arteries, to the capillaries, the venules, and then back to the heart. All right. We're going to look at this more when we look at the respiratory system. But you can see that lymphatic flow, and this is what's going through here. So let's look at some of these major vessels. You don't have to know all of them, but you should know some of the major ones. So the superior vena cava coming into that right atrium, and then obviously the inferior vena cava that you can't see. When we get to the arteries, this is the aorta. This is the ascending aorta, the aortic arch, and then the descending aorta. You have 3 branches off the aorta. Pig has two, but for humans, we have the brachiocephalic trunk or the brachiocephalic artery. Then we have the left common carotid and the left subclavian. So going to the neck, carotid, going to the shoulder, across the chest here, subclavian, under the clavicle. So this area right here in your chest, and then going the armpit area, axillary, into the brachial, which is the arm. That's kind of like the common naming system. For veins, when you get into the neck, it's jugular. All right, and I will show you that in the pig. But for the arterial system, obviously the aorta is the largest. The main portions are the ascending aorta, the aortic arch, the descending aorta. You get through the diaphragm and it becomes the abdominal aorta, so it changes names. Okay. The major branches off that arch, brachiocephalic trunk, which splits into the right common carotid, right subclavian, the left common carotid, and then the left subclavian. So you got left common carotid, left subclavian, and the brachiocephalic trunk does the right common carotid, the right subclavian. If you can remember that, you're good. You do have the aorta does branch into two common iliac arteries in the legs. We'll look at that. There is a circle, the circle of Willis in the brain, which supplies the blood to the brain. And you can look at some of these, but we're not going to go crazy. The ones that you're going to be responsible for are going to be the ones that I'm doing in the dissection. So make sure you go and look at the dissection. But you can see this descending aorta coming down, and you have the celiac trunk breaking off, which goes to the hepatic, splenic, and gastric arteries. Then you're going to break off and go to your kidneys, the renals. You are going to break off and go to the mesenteric, your superior mesenteric. Go this way. And your inferior mesenteric's going to that connective tissue that holds the intestinal tract together. Also right there at the kidneys is the gonadal artery going down into the ovaries and the testes. All right. And then you come down here and you break into your iliacs right at the hips to go into the legs. And you can see some of these branches coming off. You have specific ones you need to know, and I'll have that in your lab. All right. In terms of the venous system, this is going to return it. Again, the pathway of the larger veins usually runs right along with the arteries. So that when you'll see this with the dissection of the pig. When we dissect that pig open, you got the arteries and veins right next to each other. Okay, so that's not a problem. So you can see these major veins. One thing I do want to mention is portal veins. These are unusual. They don't transport blood directly to the heart. Basically, you're going through two sets of capillaries, and you have a hepatic portal system and a renal portal system, and the hepatic portal system, again, is unique because we want to take this blood that's coming in from the intestinal tract and send it to the liver to let the liver look at this before we send it on to the rest of the body. So basically, it'll work its way through the liver before we send it out. Some of these lifespan changes, again, I think of, as we get older, we do get cholesterol getting deposited in the vessels. We get narrowed coronary arteries. Basically, the heart muscle begins to decline. You get increase in fat tissue in the heart. The heart valves thicken. You get an increase in systolic blood pressure. You get a decrease in resting heart rate. And you get a decrease in arterial elasticity. All right, so that finishes up the heart, the cardiovascular system, and the blood vessels, and we will see you next with immunity. And You can take a look at the coronary artery disease, and take a look at that application.